Okay, let's unpack this. Imagine a future where we can precisely edit our own genetic code, not just, fix a single error, but seamlessly insert entire therapeutically relevant genes, almost like copy-pasting a missing instruction manual directly into our DNA. For anyone tracking the breathtaking pace of genomic medicine, today's deep dive is definitely for you. We're exploring a truly groundbreaking advancement that brings us, well, significantly closer to that reality. We're talking about programmable DNA integration using a new class of tools called type VK CRISPR-associated transposases, or Cas systems for short.
Yeah, and what's really fascinating here is how this research takes inspiration directly from the natural world, specifically from the ingenious molecular machinery found in microbes, of all places, to develop this powerful new approach for genomic medicine. So this deep dive focuses on a recent article, Integration of Therapeutic Cargo into the Human Genome with Programmable Type VKCST by Jason Liu and Daniela S. Aliaga-Goldsman and their colleagues. Our mission today is basically to extract the core insights from their really extensive work. We'll get into the nuts and bolts of how these compact systems work and the clever ways they've been engineered for human cells, and of course what their potential implications are for treating a whole array of genetic diseases.
Right. So you'll learn how this compact system fundamentally differs from its predecessors, the ingenious strategies researchers use to make it functional in our own cells, and how it's already demonstrating its astonishing ability to insert pretty significant pieces of DNA. We're talking even a gene to combat hemophilia into specific safe locations in our genome. This isn't just about making small tweaks anymore. It's about writing whole new chapters into the book of life, really, with an unprecedented level of precision. So if you're ready, let's dive in. So to kick us off, maybe for someone who follows the general CRISPR story, what exactly are Cas systems and what makes them such a big deal in this evolving landscape of gene editing?
Well, okay. At their core, Cas systems, CRISPR-associated transposases, are essentially RNA-guided molecular machines. You can think of them as incredibly precise biological robots, really, capable of integrating large segments of DNA, we're talking entire genes or even small gene networks, into a target genome. Now, the real game changer here, the crucial distinction that sets them apart from, say, the more widely known tools like CRISPR-Cas9, is how they achieve this integration. They do it without creating double-strand breaks in the target DNA.
Okay, and why is avoiding those double-strand breaks so significant? I thought, the whole power of CRISPR-Cas9 came from making those precise cuts.
You're absolutely right. Cas9 is brilliant at making those cuts, and it's incredibly powerful for many, many applications, no doubt. But those breaks, even though they're targeted, they can trigger the cell's own natural repair mechanisms, and those aren't always perfect. So when the cell tries to patch up a double-strand break, it can sometimes introduce unwanted insertions or deletions. We call those indels, or even larger, less predictable chromosomal rearrangements. Imagine trying to fix, like, a faulty line in a complex computer code. If you cut the wire, you hope the system reconnects it perfectly, right? But there's always a risk of a short circuit or maybe some miswiring. So by integrating DNA without inducing these breaks, Cas systems offer a much cleaner, potentially safer, and certainly more predictable way to deliver that DNA cargo. The ability to programmably deliver DNA with such high specificity, that's been a major challenge for a long time in genomic medicine, and Cas systems really represent a significant leap forward in tackling that fundamental problem.
That makes a lot of sense. Yeah, less collateral damage is definitely a better outcome when you're working with something as intricate as our own genetic code. Now, this study specifically focuses on a new variant, type VK-KOCT. How does it compare to other KOC systems, particularly the type IF cascade EKC that we've heard about before?
That's a great question, and it really highlights why this type VK is generating so much excitement. Let's break down the key differences. Okay, so type IF cascade Cas-D systems, they're derived from what are known as TN7-like elements, and they are quite complex molecular machines. They require multiple protein components specifically, several different Cas proteins, just to find their target and, kick off the process. They also contain an additional component called TNSA, which enables a more traditional sort of cut-and-paste integration mechanism. However, deploying these multi-component systems effectively in human cells has faced some pretty significant hurdles, partly because of their complexity and partly because they need specific host factors, like certain bacterial helper proteins, just to function efficiently. They're a bit like a sophisticated, maybe, but bulky multi-tool. It needs many parts to come together perfectly.
So powerful, but perhaps a bit cumbersome to use. And type VK, the star of this particular deep dive.
Exactly. Type VK case systems, on the other hand, they evolve from transposon proteins that are distantly related to TN5053 transposons. Their core advantage and why they're the focus of this groundbreaking study is their simpler composition. They require only a single Cas12k effector protein for targeting. That inherent simplicity is a major, major benefit for translating them into practical human genome editing applications. Think about it. Fewer moving parts often means less can go wrong, right? And it's just easier to engineer. Another distinction is that type VK systems generally lack that TNSA protein we just mentioned. And this has interesting implications for the types of integration products they create, which we can definitely delve into a bit later.
Simplicity sounds like a huge win for future therapeutic applications. But do these different types of KCAS systems share any fundamental components at their core?
They do, yeah. Both type IF and type VK systems share the core components responsible for the actual transposition, the physical act of moving and inserting the DNA. These include a transposase enzyme called TNSB, and AAA plus ATPase called TNSC, and a Cas recognition component known as TNIQ. It's really the Cas effector component, which is KCAS12K in the type VK system, that truly distinguishes their targeting mechanisms and, well, contributes to that streamlined design.
Okay, so the researchers hypothesized that type VK's simpler nature would make it easier to adapt for use in human cells. How did they actually go about finding these systems in the first place? And then, start to test them.
Their approach was quite innovative, actually. They started with a large-scale metagenomic discovery effort. Imagine sifting through the collective genetic material from literally thousands of uncultivated microbes found in all sorts of environments. It's like exploring an uncharted genetic wilderness. This allowed them to identify a really diverse array of type VK Cas systems that hadn't been previously characterized. It's kind of a testament to the idea that nature has often already solved the problems we're trying to tackle in the lab. So in the original research paper, the team meticulously mapped out the genomic context of one of these systems they identified, which they called MG64-1. If you look at, say, figure 1A in the paper, this blueprint revealed the characteristic arrangement of the core transposon genes TNSB, TNSC, TNQ strategically placed right alongside the Cas12k effector, plus the TRAC RNA and the CRISPR array. It also showed the distinctive terminal inverted repeats, which are kind of like molecular breadcrumbs marking the boundaries of the transposon, and a unique molecular signature, a TAA duplication right at the site of insertion. This tiny duplication is a critical clue, basically confirming that the system has precisely done its cut and paste job with the new DNA.
Right. So they uncover this fascinating diversity. What's the practical benefit of having so many different variants, rather than just like one super tool?
That's precisely why this diversity is so exciting. Their deep search revealed, as shown in Figure 1b, a significant phylogenetic tree of these Cas12k effector sequences. This wide range of discovered systems is incredibly important because it provides a much wider toolkit, you could say, or maybe a deeper genetic palette for future applications and optimization strategies. Think of it like having many different types of wrenches in your toolbox, right? Some might be better suited for different jobs, or you might find one that fits perfectly where others just don't. They also identified some fascinating conserved features within these diverse systems, things like specific motifs at the tail end of the cRNA and a distinct stem loop structure in the TRAC RNA. You can see details in supplementary figures 2 and 3. What's amazing, though, is that despite significant sequence differences between these systems, the active single-guide RNA designs, like those shown in Figure 1c for systems MG64.1 and MG64.6, still maintain these crucial conserved structural features. It really demonstrates the fundamental underlying principles of how these guide RNAs function, regardless of their specific origins.
That structural conservation, even with sequence differences, sounds really key to understanding how they work.
Okay, so once they found these diverse systems, how did they then characterize their activity and specificity in a controlled environment, like in vitro?
Right. For that, they set up very precise in vitro experiments, essentially miniature molecular reactions in a test tube. That's shown schematically in Figure 1d. They combine purified, lab-expressed Cas proteins, along with a specific guide RNA, a linear donor DNA fragment, which is the genetic cargo they want to insert, and a target plasmid library containing various PAM motifs. The whole goal here was to see if the active Cas systems would selectively integrate the donor DNA precisely downstream of a recognized PAM motif. The PAM, remember, the protospacer adjacent motif, is that short DNA sequence the CRISPR system recognizes. It acts like a molecular signpost telling the system exactly where to act.
And did it work? Did they see the integration happening like they expected? Absolutely. Their initial assays, shown in Figure 1e using PCR, confirmed successful integration for both MG64 and MG64-6. This was the first clear evidence that these systems possessed programmable sgRNA-dependent activity, meaning the integration only happened when the specific guide RNA was present and directing it to a particular spot. They then used advanced sequencing techniques, explained in Figure 1F, to pinpoint the exact PAM preferences for each system. For MG64-1, it preferentially recognized a 5-OR GTN PAM. For MG64-6, it was a 5-OR GTN PAM, where ARN indicates a weak AG preference. Knowing these precise PAM sequences is vital because it dictates where in the genome these tools can be programmed to integrate DNA.
Okay, so they know where it integrates down to the PAM sequence. How precise is that integration, specifically the distance from the PAM? That seems critical.
Remarkably precise. Their detailed analysis, which you can dig into in supplementary Figure 5b and c, showed that a staggering 90% of the integration evidence for both MG64-1 and MG64-6 occurred precisely within a very narrow window. We're talking between 57 and 67 base pairs away from the PAM. This narrow integration window is a huge advantage for therapeutic applications. Imagine trying to replace, I don't know, a faulty part in a complex engine. If you know exactly where the new part will land, within a few millimeters, it's far easier and safer than if it could end up anywhere within, say, a few feet. This level of predictability really moves gene editing from something that could be a bit random to a highly precise and therefore potentially much safer therapeutic intervention.
That's an incredible level of control. And I think you mentioned something about component swapping earlier. What's that about?
Yes, this is where it gets really interesting for future optimization and engineering, potentially. The researchers made a somewhat unexpected but highly valuable discovery. They found that even Cas12k effectors and guide RNAs from partial or seemingly inactive Cas systems, ones they found in the metagenomic data, could actually become active when paired with functional transposase components from other systems, like MG64-1. For instance, a Cas12k from a system called MG64-2, which normally wasn't active on its own, became fully functional when they combined it with the transposases from MG64-1. This modularity means scientists have significant flexibility to mix and match components, potentially creating optimized Cas systems with enhanced activity or specificity for different applications in ways maybe not possible before. It's sort of like finding that parts from different generations of an engine can be combined to create a more efficient hybrid.
Yeah.
Wow, okay. So they've identified these promising type VK-Cas-T systems, understood their precision in a test tube, and even found ways to mix and match components. The next logical step, and probably much harder, is seeing how they perform in actual living cells. Where did they begin with that challenge?
Right, the cellular context. They began, as is often done in this field, by benchmarking the systems in E. coli. It's a common and well-understood model organism. This allowed them to rigorously gauge the efficiency and specificity of these case systems in a living cellular context before tackling the significantly greater complexities of human cells. Think of it as a crucial intermediate testing ground.
And how did that E. coli experiment work? What was the setup?
For their E. coli experiments, illustrated in Figure 2a, the researchers used a clever plasmid-based setup. They engineered 3 separate plasmids. One, a helper plasmid, contained the protein-coding components of the Cas system. Another, P-donor, carried the specific DNA cargo they wanted to integrate. And a third, P-guide, expressed the single guide RNA that would direct the integration. These were all introduced into an E. coli strain. The goal was simply to observe if the Cas system would integrate the donor DNA into specific, engineered, or even natural target sites within the E. coli genome.
Okay. And what were the results in E. coli? Were they promising enough to move forward to human cells?
They were very encouraging, yes indeed. The team achieved pretty impressive integration efficiencies, shown in Figures 2b and 2c. They reached up to 80% integration efficiency at both engineered and endogenous loci within the E. coli genome, which is quite high. They even pushed the boundaries further by testing multi-locus targeting, trying to integrate DNA into multiple places simultaneously within the same cell. Even under those conditions, they observed high efficiencies up to 80% at an engineered target site and 50% at a natural endogenous intergenic region. This kind of multi-locus integration efficiency is particularly exciting for applications like synthetic biology, where you might want to insert entire metabolic pathways into a bacterial cell, for example.
High efficiency certainly sounds great, but I recall something about co-integration events when using circular plasmids. What does that mean exactly, and is that a potential issue?
That's an astute observation, and it's an important detail to understand, because these specific MG64-1 and MG64-6 systems generally lack the TNSA protein, which in other systems enables a very clean cut and paste. When they used a circular plasmid as the donor DNA, they saw two main types of integration outcomes. About 20–30% of the time, they saw clean, single integrations of just the intended cargo, which is great. However, the other 70–80% of events were these co-integration events. This means that along with the intended gene or DNA fragment, additional parts of the circular plasmid backbone itself were also integrated into the genome. Now, while that's not ideal for all applications, understanding this mechanism is crucial for refining the system. It's not necessarily a deal breaker, especially if you can switch to linear donors later on, which, spoiler alert, they did explore.
Right. And what about off-targets in E. coli? That's always a big concern with any gene editing tool, regardless of the organism, right? Safety first.
Absolutely. And this is a really positive finding from their E. coli work, speaking directly to the potential safety profile of these new systems. Previous studies with other type VKST systems, like a well-known one called SHAST-TCAS, had reported pretty significant off-target integration rates, sometimes as high as 40–60%, which is, frankly, quite concerning. However, in their multiplexed E. coli experiments, these researchers observed fewer than 7% off-targets across all conditions tested. This is a substantial improvement and strongly suggests that these newly identified type VKH systems possess a much higher level of inherent specificity, which is precisely what you want when you're designing therapeutic tools.
That's a huge step towards safety and predictability.
Yeah.
Okay, so now moving from bacteria to human cells, that introduces a whole new set of challenges, especially getting these bacterial components into the right place in the nucleus and making them work there. How do they tackle that nuclear localization problem?
That's the major hurdle, exactly. Bacterial gene editing tools aren't naturally equipped to function in our eukaryotic nucleus. They basically need a special passport. The standard solution, and what they used, involves fusing nuclear localization signals, or NLS tags, to the Cas proteins. NLS tags are short amino acid sequences that act like a cellular zip code, directing proteins to the nucleus through the cell's own transport machinery. The team undertook a really meticulous, systematic optimization process. They fused NLS tags to both the front N-terminus and back C-terminus of each Cas protein, Cas12K, TNETQ, TNSC, and TNSB, to find the optimal configuration for each one. This detailed trial and error, which is nicely summarized in Table 1 of their paper, detailing the optimal NLS tag orientations, was absolutely crucial for successfully guiding these bacterial components into the human cell nucleus. It really shows the dedication to incremental improvements that leads to these kinds of breakthroughs.
So after all that optimization detailed in Table 1, did the NLS tags work? Did the proteins actually get into the nucleus as intended?
Yes, their imaging studies, shown in Figure 3A, confirmed this. Most of the NLS tag proteins efficiently translocated into the nucleus of HEK293T cells, a common human cell line used in research. There was one initial exception, TNSC, which was a bit reluctant. But interestingly, they made a key observation regarding TNSC. They found that co-expressing TNIQ, along with TNSC, significantly improved TNSC's nuclear localization. This strongly suggests a direct interaction between these two components, which is fascinating, and actually aligns with structural observations from other Cas systems. It's like they form a little buddy system, a mini-complex, that helps usher TNSC into its proper cellular compartment.
Okay, but just getting them into the nucleus isn't enough, is it? They also need to be active once they're there. The cellular environment is very different from bacteria.
You're absolutely right. That's only half the battle. Simply localizing the components wasn't sufficient to restore full activity in the nuclear environment. To rigorously test this, they devised a clever method, shown schematically in Figure 3B. They created nuclear extracts from cells expressing the NLS tag Cas-E components, and then tested the activity of these extracts in vitro. And initially, they found that even with proper nuclear localization, the NLS-tagged Cas12K and TNEQ were surprisingly not fully active. It suggests the human nuclear environment must be different enough, perhaps lacking certain cofactors or having inhibitors, that it hinders these bacterial enzymes.
Okay, so the proteins are in the nucleus, but they're not working properly. That's a classic scientific puzzle. How did the researchers crack that code? What was their aha moment that got the system up and running?
This is truly a brilliant piece of engineering, and yeah, where the deep dive gets really exciting, as Figure 3C shows. To overcome this hurdle, they took inspiration from advancements made in optimizing other CRISPR enzymes for the nuclear environment. They decided to fuse Cas12k and TinyQ with chromodomain or processivity factors. Specifically, for Cas12k, they used a small protein called SOTHO7, known to help proteins bind DNA. And for TinyQ, they used either H1 core or HMGN1, which are involved in chromatin structure. What they found was remarkable. These fusions dramatically activated the Cas system components that had been extracted from the nuclear environment. These additional domains likely help by stabilizing the Cas-T components within that complex nuclear environment, perhaps by helping them bind to DNA more effectively, or maybe by compensating for missing human host factors that are needed for their activity. It's a really clever way to essentially trick this bacterial machinery into working efficiently in a completely different eukaryotic cellular context.
That's truly innovative thinking. So, with the components now active and in the right place, did they also observe a favored direction for integration? Because that sounds like it would add another layer of predictability, which is always good.
They absolutely did, and it's another significant advantage. Their in vitro experiments, especially when working with rare targets or the complex human DNA, detailed in supplementary figure 13AB, revealed that these key ST systems strongly favor a single integration orientation, specifically what they called forward. This means that when the DNA cargo is inserted, it consistently goes in one specific direction relative to the target site. This predictable directionality is a huge benefit for many therapeutic applications, as it allows for much more precise control over how the inserted gene will be expressed and how it might interact with the surrounding genomic landscape. You definitely want to make sure your new instruction is oriented correctly so the cell can read it properly.
Definitely. And to really push the boundaries then, they started testing integration directly into human genomic DNA, but focusing on multi-copy loci first, right? Why target those multi-copy sites initially, and what did that prove?
Targeting multi-copy sites was a strategic and very smart initial step. It basically simplifies the initial search for successful integration events within the sheer vastness of the human genome. Imagine trying to find a single specific needle in a giant haystack versus finding a cluster of thousands of needles. Much easier, right? They aimed for these high copy elements like line 1A 3 minutes, SVA, and RV elements. As Figure 3D highlights, these are present at roughly 10,000, 1,000, and maybe 4 copies per haploid human genome, respectively, in the cell lines they were using. That immense difference in target availability compared to, say, a unique single-copy E. coli site vividly illustrates why multi-copy targets made sense for early validation in the human context. And their efforts paid off. As shown in Figure 3E, they successfully achieved in vitro integration into these high-copy elements, line 1A 3-minute SVA and HERV, covering a huge 3-order-of-magnitude range of target concentrations. This was a critical proof of concept. It demonstrated that the system could indeed function effectively with human DNA as a target, laying crucial groundwork for their ultimate goal: integration into single-copy, therapeutically relevant sites later on. It showed the core machinery was working against the complex backdrop of our genome.
Got it. So with the in vitro success demonstrating the system's potential to work with human DNA, the next big leap was seeing if these Cas systems could integrate DNA directly inside human cells, not just in a test tube with extracted DNA. How did they set up those crucial initial in-cell experiments?
Right, moving in cellulo. For those initial experiments directly within living human cells, they started with a fairly common approach called a dual plasmid system. You can see this on the left side of Figure 4A. This setup involved 2 separate plasmids. One plasmid expressed all the necessary Cas-TIC components, including a key host factor they identified called S15, which is tagged with an NLS to ensure it got into the nucleus. The second plasmid carried the specific guide RNAs designed to target multiple line 1A3 sites within the human genome. This allowed them to meticulously control the delivery of each piece of the puzzle.
And what were the immediate results? Did they actually see signs of integration happening in those cells?
Yes, the results were definitive and very exciting. As shown in Figure 4B, their analysis showed successful detection of target donor junctions at multiple line 1A3 targets in human HEK293T cells. This was the first direct, compelling evidence of type VK highest activity actually happening inside living human cells. A huge step. And crucially, this integration occurred exclusively in that favored forward orientation we discussed earlier, reinforcing the predictability of the system even in the cellular context. This was a massive milestone, really, moving from controlled test tube reactions to the dynamic environment of a human cell.
That's a huge step forward, absolutely. But managing multiple plasmids can be a bit cumbersome for real-world applications, can't it? Did they try to simplify the delivery system even further?
Absolutely. Simplification is often key for therapeutic translation. To streamline delivery and reduce design constraints for future applications, they ingeniously developed an all-in-one plasmid, shown on the right side of Figure 4a. The single plasmid was engineered to contain all 5 protein encoding components: Cas12K, TNSB, TNSC, TNAQ, and S15, cleverly separated by molecular spacers called IRS and 2A elements. You can think of these like special markers that tell the cell's machinery, okay, make this protein, then make the next one, even though they're all on one long genetic message. This single plasmid also included the single guide RNA expression cassette, so everything needed in one package. This all-in-one approach significantly simplifies the delivery process, making it much more practical for future use. And importantly, they successfully validated human genomic integration into those high copy line 1A, 3R elements using the simplified construct. Supplementary Figure 16a, proving its efficacy.
It really sounds like they're refining the system for practicality. What about boosting the efficiency even further? I remember CLPX was mentioned earlier as a bacterial helper protein that might help.
Yes, good memory. CLPX, a bacterial protein, and AAA plus ATPase, had previously been shown to improve integration for other different Cas systems, the cascade type. Researchers hypothesized it might help somehow resolve the integration events, just make the process more efficient overall. So they tested whether CLPX could similarly enhance type VK Cas efficiency. And the results, noted in Table 1 and shown in Figure 4e and Supplementary Figure 16b, were encouraging. Both N and C-terminal NLS fusions of CLPX significantly enhanced integration efficiency at that high copy line 1A 3' locus. This strongly suggests CLPX plays a beneficial role in type VK-Cas activity as well, likely by aiding in the resolution of integration events, perhaps by helping the system handle the complex DNA structures involved in the integration process. Another useful knob to turn, basically.
Right. Okay, so with all these optimizations and refinements, the all-in-one plasmid adding CLPX, were they finally able to hit a really important single copy target site, something like what's often referred to as a safe harbor locus in the human genome?
This was indeed the critical test for therapeutic relevance and a major turning point in their research. They targeted AAVS1. AAVS1 is a very well-known single-copy safe harbor locus in the human genome. It's extensively studied and used in human therapeutic applications precisely because genes inserted there are thought to be much less likely to disrupt essential cellular functions or accidentally activate problematic genes like oncogenes. It's a bit like finding the perfect designated parking spot for your new genetic instruction, where you know it won't cause any traffic jams or block driveways.
Makes sense. And what did they find at AAVS1? Was the integration successful there?
The results were highly successful, a real validation. With the all-in-one plasmid, the donor plasmid carrying the cargo, and the crucial addition of CLPX, they successfully detected the integration of a substantial 2.8 kilobase donor fragment at 2 distinct AAVS1 sites, which they designated T1 and T2, shown in Figure 4c. For AAVS1T1, they even provide a compelling visual confirmation through Sanger sequencing, presented in Figure 4D. You can literally see the sequence trace showing the integration event at the molecular level. This wasn't just detection, it was clear, undeniable evidence. The quantitative analysis using next-generation sequencing, NGS, really drove home the efficiency point. As shown in Figure 4e, the addition of CLPX dramatically enhanced editing rates by a remarkable 5-fold. This resulted in over 1% average integration at the AAVS1T1 site. Now, 1% might not sound huge initially, but for gene editing, particularly for therapeutic applications where even a small percentage of corrected cells can have significant clinical benefits, that's a very promising efficiency.
That's incredibly impressive, especially at a single copy safe harbor. How did their optimized MG64-1 Cas system compare to other type VK systems, like the well-known SH/T, when targeted to this crucial AAVS1 site?
That's a key point of differentiation and really a testament to the power of their metagenomic discovery approach. As shown in Figure 4F, under the exact same optimized conditions, their MG64-1 Cas system achieved a solid 2% targeted integration at AAVS1T1. In stark contrast, the previously well-characterized S. Hoffmanni type VK Cas-T, or SH Cas-T, showed no detectable reads for target-specific integration, 0. This powerfully highlights the superior performance of the metagenomically derived MG64-1 system compared to its known counterparts, at least under these conditions. Furthermore, their analysis confirmed no unexpected modifications like indels. Those insertions or deletions were detected at the target site, Supplementary Figure 17, which is absolutely crucial for safety.
That's a massive performance advantage. But okay, for any gene editing technology, particularly one aiming for clinical use, addressing specificity and demonstrating minimal off-target activity in human cells is paramount. How did they tackle this critical concern, especially given the, well, sheer vastness of the human genome?
This is indeed the make-or-break question for clinical applications, you're right. Detecting potentially rare off-target integration events in the vast, complex human genome is a significant technical challenge, especially getting the sequencing depth needed to find them reliably. To meet this challenge, the researchers developed a novel, quite sophisticated deep sequencing method, diagrammed in Figure 4G. This method was specifically designed to accurately identify complete integration events by enriching for both the left end, LU, and right end, RE, junction sites across the entire human genome. The schematic shows how they designed it so that reads aligning to both LE and RE junctions and confirming the presence of that characteristic target site duplication, TSD, are essential for identifying a complete donor integration. This rigorous approach ensures they weren't just detecting partial insertions, which might not be functional, but full, potentially functional integration events.
Okay, so they're not just looking for partial integrations, but complete ones, which makes sense. And did this method confirm the precision of the on-target integration they'd already observed at AAVS1?
Yes, it provided even further validation of their precision at the intended site. Their data, shown in Figure 4H, clearly demonstrated the convergence of the AAVS1T1 on-target reads. The left end and right end reads aligned precisely with the expected overlap distance due to that target site duplication. This definitively confirmed the complete and precise integration of the donor at the intended location. This validation gave them high confidence that their method was robust enough to accurately and comprehensively evaluate potential off-targets across the entire human genome, not just focusing on the intended spot.
Okay, so the moment of truth. After all this meticulous work and the development of this new assay, what did they actually find regarding off-target locations in human cells?
The results provide a nuanced and really important picture. First off, most of the detected off-target events were actually plasmid-to-plasmid integration events. What this means is the KST system, instead of always inserting DNA into the human genome, sometimes inserted DNA from one plasmid into another plasmid that was also present in the cell at high copy numbers. These included insertions within the helper plasmid itself, for example, in the H1 core chromodomain region, the sgRNA promoter, and other regulatory elements on the plasmids. This suggests that these events might be more frequent simply due to the higher availability of plasmid substrates within the cell, essentially an easier target environment for the KST system compared to the complex chromatin of the genome.
That's a very important distinction, yeah. It means the system isn't necessarily running wild within the human genome itself. So what about off-targets directly within the actual human chromosomes? Were there any, and where did they land?
While rarer, they did observe up to 8 distinct human chromosomal DNA off-target sites when a guide RNA was present. Now, crucially, 5 of these sites were reproducible across multiple experimental replicates, as shown in the Venn diagram in Figure 4I. This reproducibility indicates they aren't just random one-off occurrences, but rather represent a consistent, albeit low-frequency pattern of off-target activity. And these reproducible off-targets tended to cluster in specific genomic hotspots. These hotspots were found in critical functional regions, such as regulatory elements like the EF1A1 and FTL promoters, and also the 3-or untranslated region, UTR, of the HBB gene, beta-globin. This clustering in functional regions tells us something important about their nature, perhaps related to accessibility or local chromatin structure.
Okay, but were these off-targets driven by the guide RNA sequence itself? Like what sometimes happens with Cas9, where a partial match leads to an off-target cut?
This is a critical and actually quite reassuring point from their analysis. They were not driven by spacer homology, meaning these off-target sites had many mismatches with the guide RNA sequence. This indicates that the guide RNA wasn't accidentally matching another site in the genome due to sequence similarity, which is a common concern with other systems. The implication here is that these off-targets are likely controllable, probably by further engineering of the Cas-T components themselves, or maybe by carefully tuning the expression levels of key proteins like TNSC. That's a strategy that has shown success in reducing off-targets in previous studies with other Cas systems like Change-Cas. This suggests that while these off-targets exist and need to be addressed, they seem manageable through optimization, which is a very positive sign for clinical development. It points towards the system's tunability rather than an inherent, unavoidable lack of specificity based on the guide sequence.
All right, so controllable off-targets, that's key. Okay, so they've shown precise, efficient integration in human cells, even at a safe harbor site. And they've begun to characterize and understand the off-target activity. What about the versatility of this system? Can it work in different human cell types? And maybe with different kinds of DNA donors? Because those are crucial factors for real-world application.
Excellent question, as versatility is absolutely key for any broad therapeutic application. You're right, the initial experiments in HEK293T cells use plasmids with an SV40 origin. Now, this origin allows the plasmids to replicate within those specific cells, which can potentially boost both the expression of the KRAS-ST components and the availability of the donor DNA, and that can lead to higher observed integration efficiency. But for broader therapeutic deployment, you absolutely need systems that are effective in cell types that do not have this replication capability, which is most cells in the body.
So how did they test that independence from plasmid replication? Which cell types did they use?
They rigorously tested KST in cell types that do not express the SV40 large T antigen, meaning any transfected plasmids would simply not replicate within those cells. They used two important cell models: immortalized myelogenous K562 cells, which are often used in blood disorder research, and liver carcinoma Hep3B cells, which are obviously relevant for potential liver-directed therapies. The results, shown in Figure 5A and 5B, were clear. They observed successful targeted integration at the AAVS1 safe harbor site in both K562 cells and Hep3B cells, with efficiencies reaching up to 0.3%. This is a really vital finding because it confirms that the integration process itself is not dependent on plasmid replication. That's a huge step towards clinical translation and wider applicability across various clinically relevant cell types in the body, where cells often are actively replicating introduced plasmids.
That's a significant barrier overcome for practical use, definitely. And what about the form of the donor DNA itself? We've mostly talked about circular plasmids, but I understand linear DNA donors could be a big advantage for manufacturing safety too.
You're absolutely right. This study also explored another important frontier: using a linear DNA donor instead of the circular plasmids, as shown in Figure 5C. This is highly significant for clinical translation because linear donors offer several potential advantages. They enable bacteria-free manufacturing, which makes them easier to scale up for production and potentially safer for delivery to patients, as you avoid any lingering concerns about bacterial plasmid backbones or antibiotic resistance genes. The results showed that integration at the AAVS1 locus was indeed detected with a linear donor, albeit at slightly lower levels compared to the plasmid-based experiments in these initial tests. But this clearly validates the use of type VKKST for programmable genome editing, even with gene-size cargo, using a more clinically relevant and streamlined delivery method. It means you don't necessarily need the whole plasmid, just the piece of DNA you want to insert.
This really sounds like it's getting closer and closer to real-world applications now. Did they actually test integrating a specific therapeutically relevant gene? Something for a known human disease?
Absolutely, and this is truly a major proof of concept for the entire system, moving beyond marker genes. They chose hemophilia B. Hemophilia B is a serious inherited bleeding disorder caused by mutations in the factor IX, FIX clotting factor gene. It's considered a prime target for gene therapy because stable, lifelong integration of a functional histogene offers the potential for a one-time, potentially curative treatment, eliminating the need for frequent, costly protein infusions. So they carefully constructed a factor IX donor plasmid, shown schematically at the top of Figure 5D. This plasmid contained the full human fixed gene, which is 1,383 base pairs long. That's a substantial piece of DNA driven by a strong liver-specific promoter, chimeric PAAT, amounting to a total of 3.6 kilobases of therapeutic cargo. So this wasn't just a small marker gene, it was a full, functional therapeutic gene cassette.
Wow. And were they able to integrate this large therapeutically relevant gene into human cells at the AAVS1 safe harbor site?
Yes, the results were highly successful and really demonstrated the true potential of this technology for gene therapy. As measured by NGS and shown in Figure 5E, they achieved successful targeted integration of the entire FIX cargo into the AAVS1T1 site, with over 1% integration efficiency. This is a monumental step. It directly shows the system's capability for real-world therapeutic applications. Imagine the impact of reliably inserting a functional gene to correct a severe genetic disorder like hemophilia, potentially offering a long-term cure. It's truly transformative potential.
That's incredible. 1% efficiency for a full gene like that is really something. And were there ways to make that integration even more efficient, especially for a therapeutic gene like factor IX where maybe you need higher levels?
They did explore that, yes. And it's another clever optimization strategy they demonstrated. They found that simply increasing the expression level of the guide RNA itself could significantly boost efficiency. By designing a donor construct that contained two identical guide RNA cassettes, essentially providing more copies of the address label to guide the machinery,
Hmm.
shown at the bottom of Figure 5D, they observed a near linear increase in FIC-hex integration efficiency. As shown in Figure 5F, this achieved up to a threefold increase compared to the single guide RNA construct. This provides a clear and relatively straightforward path for further optimizing the system's efficiency for therapeutic applications, simply by providing more of the guide RNA to direct the integration process. It's a fundamental principle, really. More specific instructions can lead to more successful operations.
Makes sense. And finally, for a therapeutic gene to truly work, the entire donor DNA needs to be integrated correctly, right? Not just pieces of it. Did they confirm complete integration of the factor IX gene? Because a partial gene probably wouldn't produce a functional protein.
That's absolutely crucial for producing functional proteins and delivering any kind of lasting therapeutic benefit. You need the whole message. They rigorously address this by analyzing the integration efficiency detected from both the left end, LE, and right end, RE, target to donor junctions. What they found, presented in Figure 5G, was that the relative integration efficiency measured from these two ends approximated a 1.1 ratio. This is strong evidence indicating that a vast majority of the integration events are indeed complete, meaning the entire 3.6 kilobat factor IX gene cargo was successfully inserted from one end to the other. This demonstration of complete and precise integration is a robust validation of type VKTC's potential to deliver functional therapeutic genes, ensuring the cell receives the full, correct instruction manual.
Wow. This deep dives into type VK-Cas systems truly paints a vivid picture of the future of genomic medicine. I mean, from discovering these powerful tools in the hidden world of uncultivated microbes, to meticulously engineering them for precise, safe, and efficient integration in human cells, the advancements are just astounding. The ability to precisely insert a full therapeutic gene, like factor IX, into a safe harbor site, doing it in various cell types, even using linear DNA, that feels like a monumental leap. It's really not just about fixing a single letter in our genetic code anymore, is it? It's about adding entire functional sentences and paragraphs to the genome.
Indeed. This work lays absolutely crucial groundwork, offering what looks like a genuinely transformative platform for genomic medicine. It's very exciting. The observation of those off-target hotspots not being driven by guide RNA homology, while certainly needing further optimization, it actually offers a clear path forward for refining specificity, likely through fine-tuning protein interactions or maybe just optimizing expression levels, perhaps, of TNSC. The flexibility shown by component swapping and the demonstrable impact of factors like COPX suggest this is a highly tunable system. It seems capable of being adapted for a wide range of therapeutic needs. This continuous pursuit of understanding and enhancing these systems, like the work shown here, promises truly transformative applications for genetic diseases. It moves us closer to therapies that are not just effective, but potentially enduring, precise, and maybe, just maybe, curative.
So, as you reflect on this incredible progress, maybe consider this provocative thought. If we can program living cells to precisely integrate entire therapeutic genes, what other complex biological instructions might we one day be able to, copy-paste directly into our genome? Can we fundamentally reshape how we approach not just rare genetic diseases, but also common illnesses, perhaps aging, or even enhance certain human capabilities? The possibilities seem truly boundless, and this deep dive really shows we're getting closer to that future every day.